Hi, so in this video, I want to talk a little bit more about directing proteins to the nucleus. So you might be asking yourself, okay, that's interesting. That's how the cell does it. Is there any reason why we might want to know that? Well, actually, it's pretty important for how we perform biological experiments. So if you have an experiment you want to do on human cells, so like say for example this one right here, um, if we just sort of bathe that cell in some sort of protein, or even if we insert a gene into that cell to create a protein, that protein is going to be created here in the cytosol, or if we were to just sort of cover it in that, it would just be right here, and maybe it gets into the cell. It's not going to get into the nucleus. So one of the things that is helpful when we're doing research is that we can actually create proteins. So get the nuclear, the sorry, the DNA sequence of a protein, put it in the DNA of a cell. The cell will create that protein out here in the cytosol, and if we just add a tag that directs it, say, for example, to the nucleus, then the protein itself, or the cell itself, will import it back into the nucleus. So we can do much more sophisticated studies with recombinant DNA by creating proteins that were not originally intended for the nucleus of a given organism, or for that matter, for the ER, or for that matter, for the mitochondria of an organism, and have them actually go to a different location than perhaps they were originally used or originally intended to go. And that's a powerful way for us to do different sorts of studies.